Hello, hello. Good evening. Hello. Am I audible? Hello. Am I audible? Hello, hello. Hello. Good evening. Welcome to GoScore Learning live session. We are the fastest growing edtech in GCC and we make learning easy for our students. And our aim is to provide education to all students. Okay, so again, myself Amala Anjos, and I'm back here with molecular basis of inheritance part 2. So what we were discussing in part 1? Yes, it was about RNA and DNA, right? So today we will be discussing the further topic of this chapter, that is about how DNA becomes the genetic material. What are the different studies to understand, or what are the different experiments over done to understand how the genetic material was discovered, or we can say how the genetic material DNA is not as DNA is done with different methods of experiment. So here we have to discuss with three different experiments to reveal that DNA is the genetic material of organisms. Okay, so first one, it will be Griffith experiment. Later on, we'll study the second experiment with MacLeod and McCarthy. Then third experiment, we'll go with Hershey and Chase experiment. Okay, these are the three different experiments that we have to learn about. So what is the first experiment? The first experiment was done by Griffith to understand what is the genetic material. Okay, so last class we have studied that DNA is the genetic material, but no one knows that exactly what is the actual genetic material, whether it is protein or DNA or RNA. There are different types of components inside the body, and nobody reveals that DNA is exactly the genetic material. Okay, so the first prior studies, which is all over the world accepted, is with Frederick Griffith, and let's see what is that experiment. Okay, so moving to our first topic, that is experiment done by Griffith. What is the Griffith experiment? Okay, so we have to discuss it with. So he used an organism, and that organism is Streptococcus pneumoniae. Okay, the organism is Streptococcus pneumoniae. That means this organism is the organism which causes a particular disease known as pneumonia, and he used two different strains of this bacteria. Okay, for his experiment, he has chose this particular organism, and different types of bacteria are there, right? And all these bacteria will show different types of strains for that. Okay, so here he chose two different strains for his experiment, that is R strain and S strain. Okay, R strain and the S strain was the strain that he used for his experiment. What is actually R strain? We will call R strain as rough strain. Okay, we'll call it as rough strain. R strain as rough strain, and the peculiarity of the strain is they are non-virulent. Okay, they are non-virulent means if this bacteria is entering into the body of any organism, they won't cause pneumonia. Okay, so we'll call it as non-virulent organisms. Okay, they are non-virulent organisms. Next, he chose the bacterial strain as S strain. Okay, S strain, or actually it is, we will call it as smooth strain. Okay, what strain it is? Smooth strain. What is the peculiarity of the smooth strain? The smooth strain means if you are considering a bacteria if you're considering a bacteria, the surface of the bacteria is made up of a smooth polysaccharide. Okay, the surface of the bacteria is made up of a smooth polysaccharide. Since the smooth polysaccharide is there over the surface. Okay, this is a smooth polysaccharide, and since this polysaccharide is present, this bacteria will be virulent. That means if it is entering into an organism, if this particular strain of bacteria is entering into a microorganism or entering into a different organism, this will cause pneumonia that will infect the organism and cause pneumonia. So that is the difference between R strain and S strain. If you are looking into non-virulent rough strain, it will be like this. That means there is no polysaccharide coating over the body of this bacteria. Okay, so you understand what is the difference between the two different strains, right? So what is the organism that he chose? He chose the organism as Streptococcus pneumoniae, and there are two different strains, that is R strain and S strain. R strain is rough strain and S strain is smooth strain. What is the speciality of smooth strain? It has a polysaccharide coating over there, and if smooth strain is entering into an organism, it will cause pneumonia, and if R strain is entering into the organism's body, it won't cause any kind of virulency. Okay, so this is the bacteria he chose for his experiment. So what is his experiment? Okay, let's see what is his experiment. So he chose the organism as mice. Okay, he chose the organism as mice, and what he has done? This mice was injected with R strain. Okay, the mice was injected with which strain? R strain. So when mice was injected with R strain, what happens? See, I told you that R strain is rough strain and they are non-virulent. Since it is non-virulent, we can say mice lives. Okay, mice lives. So second one, if you are considering, this is the first experiment he has done. Then second experiment he has done with another mice. Okay, he took another mice. Then what he has done? He has injected the S strain. Okay, what he has injected? He has injected the S strain. Okay, so when he injected this S strain, what happens actually? This S strain is virulent. S strain can be said as virulent. It causes virulency. So if S strain is entering into the mice, what happens? That causes pneumonia and the mice die. Okay, mice die. Okay, this is the prior experiment. And what happens if this organism is heat killed? That is the next step. Okay, so heat killed organisms. Okay, heat killed strains of S and R strain. Okay, so what he has done? He has taken what we can say, if you are considering this as a test tube. Okay, this as a test tube and this as a bacteria. Inside this bacteria, and what he has done? He has killed the bacteria with heat. Okay, he has killed the bacteria with heat. So the bacteria will be heat killed bacteria. Okay, so next experiment. What is this next experiment? He took the heat killed. Okay, the third experiment was with mice. He took a mice, another mice. Okay, then what does he do? He treated it with, or he injected this mice with heat killed R strain. Okay, R strain. Then what he observed? That means there is nothing. Okay, R strain. R strain is actually non-virulent. If it is heat killed, then also it is non-virulent. So we can say the mice lives. Okay, since it is non-virulent, mice lives. Then what has he done with the next one? He took the mice. when he treated or injected it with heat-killed S strain. Okay, which strain? Heat-killed S strain, and he observed for many times, and finally he got the result as mice lives. Why mice lives? Why mice lives? See, this strain was heat killed. Okay, what he has done? He has heat killed the strain. So all the components which were present inside the heat killed strain was killed. Okay, so there is no virulency. There is no virulency expressed. So we can say the mice lived. Okay, so if you are considering the next one, next process or next step, that is very important. The fifth experiment with mice. Okay, what he has done? He has taken a mice. Okay, he has taken a mice. Then he adds some R strain, live R strain, or we can say simply R strain. Okay, simply R strain. Then with heat killed S strain. Okay, so what is the probable result? What we are expecting? See, this R strain, if we are considering R strain is non-virulent. Okay, if you are considering R strain is non-virulent, right? R strain is non-virulent. Okay, if you are considering heat-killed S strain, if you are considering heat-killed S strain, this heat-killed S strain are also non-virulent, right? So, if you are injecting this R strain along with heat killed S strain, what will be the expected result? The expected result will be mice lives. Since it is non-virulent, mice will be living. Okay. So, but what did he observe? He observed the result as mice died. Okay, he observed the result as mice died. Okay, that means something which is present in heat-killed S strain. Okay, something which is present inside the heat-killed S strain transforms the R strain. Okay, transforms the live R strain into S strain. Okay, understood? Understood? Clear? That means, once again I will repeat, heat-killed strain, as I said, it is non-virulent. But when it comes in contact with R strain, okay, when it comes contact with R strain, what happens? Some elements which is present inside the heat-killed S strain changes the R strain, changes the normal R strain, okay, changes the normal R strain into S strain. Okay, we have learned that S strain is actually virulent. Okay, S strain is actually virulent. So this particular virulency will cause the death of the mice. Very simple. Okay, it is very simple. You just want to understand the concept. That's it. Okay, that is the Griffith experiment, and he suggests that a transforming principle, that something which is present inside the R strain converts it into S strain. So that what happens? It converts the S strain as virulent strain. Then mice died. Okay, so this was his experiment. Let's see what is the conclusion for his experiment. Okay, what is the conclusion for his experiment? We can look over that. If you are considering the conclusion for this experiment, see, R strain bacteria transformed by heat killed S strain. Okay, that means inside the heat killed S strain there was something present, and that something changes the heat killed S strain into R strain. Okay, so transforming principle, that is transferred from heat killed S strain, which enabled the R strain to synthesize a smooth polysaccharide coat, which cause virulency. Okay, which cause virulency, and that transforming principle, actually it was the genetic material. Actually it was the genetic material. So if you are considering Griffith experiment, he understood that there is something, and that something was known as the transforming principle. And later on, he was not able to understand what is the biochemical nature of the genetic material. That means in his experiment, he doesn't define about his biochemical nature of the genetic material which is present or which causes the transformation of heat killed strain into smooth strain. Okay, got it? Clear? Clear about Griffith experiment? So this is all about his experiment. Then finally we will be moving to the next experiment, which is again in search of the genetic material. Okay, in search of the genetic material, that is about Oswald Avery, Colin MacLeod and Maclean McCarthy experiment. Okay, you have to learn the names of this scientist. Okay, what are the three names? Oswald Avery, Colin MacLeod and Maclean McCarthy. Okay, so what does they have done with this particular or what was their experiment? Okay, see, we have learned different experiment or we have just learned about Griffith experiment, right? So they modified the Griffith experiment and they have done the experiment to determine the biochemical. Why? In Griffith experiment, they were not knowing the biochemical nature of the transforming principle. Okay, later the scientist is continuously discovering on what is the genetic material and what are the peculiarities of the genetic material. Still now it's continuous, right? So they were also doing the experiment with different mechanism, and there they have reworked this Griffith experiment. Okay, so what has actually done with this experiment? Let's see. Okay, so you know that smooth bacteria, okay, smooth bacteria or a strain will be causing the virulency. Smooth strain or a strain. Okay, smooth strain or a strain will be causing virulency. Okay, so what they have done? They have separated the different components of this strain. So see, they have different components as DNA. Okay, then they have RNA. Okay, then they have protein. Okay, so they have different components. The smooth strain has different components. And what are the components? DNA, RNA, and protein. So after that, what they have done? They have added this into a set of bacteria which is having R strain. Okay, they have added this into a plate which where it grows R strain, where they grows R strain. Okay, where they grows R strain. So see, the smooth strain is having virulency, right? So if it is added with R strain, what happens? That virulency will be causing towards this DNA or R strain. So only the transformed DNAs, transformed bacteria were present on this set of petri dish where DNA was actually transferred. Okay, so again they have done the experiment. Again they have done this experiment with another different molecules, that is they have added different components like DNAs, okay, DNAs, then RNAs, and then finally proteins. Okay, what is the speciality of DNAs? What is the speciality of DNAs? The speciality of DNAs. DNAs is an enzyme, right? So this DNAs is capable of removing DNA from a sample. Okay, what does they do? They remove DNA from a sample. Okay, so what is the function of RNAs? RNAs will remove the RNA from the sample. Okay, if a particular sample is taken and if it is RNAs is added, then the RNA present in that particular organism or particular sample will be removed. What is the condition of protease? All these are enzymes. And what is the function of this enzyme? Protease will be removing the protein. Okay, what they will be removing? They will be removing protein from the sample. Okay, so they use three different enzymes. What are the three different enzymes? It is DNAs, RNAs and protease. Okay, so what happens if this, see, this is a tube which contain a strain bacteria. Okay, it contains a strain bacteria, and they add, and what they have add? They have add DNAs. DNAs. Okay, so what happens here? All the bacteria's DNA will be removed. All the bacteria's DNA will be removed. So, if this particular, after addition of the DNAs, if it is added to mice, okay, if it is added to mice, what will be the result? So, there is no DNA. So, there is no DNA. Think that DNA is causing the virulency. So when it was added with the mice, there is no DNA, right? So when it was added with mice, what happens? Actually, the mice leaves. If DNA was present, what was the result? What was the result if the DNA was present? If the DNA was present, the mice could have been died. Okay, but see, since we use what they have used as the enzyme, they have used DNAs as the enzyme. Since DNAs was used, all the DNA molecule which is present inside the bacteria was removed. Okay, after this, what happens? It was then given to the mice. The mice lived. Okay, then second one. Next one, if you are considering the next case, they have taken the bacteria. Okay. Okay, see. This is a set of another clade where bacteria was added. Okay. And if you are considering what was used, RNAs was used. What was used here? RNAs was used. So what happens here? All the RNA molecule which is present inside the organisms was removed. Okay, so if this RNA, see, RNA is not the genetic component, right? So if you are adding this RNA will be removed, but still what is there? DNA is there. So if you are adding the smooth strain of DNA or smooth strained bacteria, Streptococcus pneumoniae, into a mice, what happens? Okay, so if you are adding this into a mice, if you are adding this type of bacteria into mice, since there is DNA, this mice will be dead. Okay, mice will be dead. Okay, understood? Clear? The same process will be taking place for the protein too. Okay, they have taken a dish, they have grown the bacteria. Okay, they have grown the bacteria. They have grown the bacteria inside. Okay, they have add what they have add. They have add protease. Okay, into this they have add protease. Okay, what they have add? They have add protease. So if protease is added, as I have told, all the protein molecule which is present inside the organism will be removed. Okay, so exactly what is there inside? Inside the bacteria, DNA is present. So if DNA is present in the smooth bacteria, smooth bacteria, what will be the result? Okay, then if it is injected to mice, since there is DNA present, what happens? Mice seems to be dead. Okay, mice seems to be dead. So they define that, they particularly says that which is the genetic material? DNA is the genetic material which makes the changes of that particular heat-killed S strain or R strain. Heat-killed S strain transform the R strain into S strain. Okay, clear? Clear? Is it clear? Okay, let's see the conclusion for that. Okay. So, what was the conclusion for their experiment? DNA from heat-killed S-strain bacteria causes our bacteria to transform. Okay, and what is the next? When protein digesting enzyme, protease, and RNA digesting enzyme, RNase, is mixed with heat-killed S bacteria, transformation of R bacteria to S bacteria takes place. Okay, that means there is a transformation of the R strain into S strain. Okay, so if digestion of DNA with DNase is taken, it inhibits the transformation and suggests that DNA causes the transformation. Okay, so finally what they have done, they concluded DNA as the genetic material. What they have concluded? They have concluded that DNA as the genetic material. Okay, again an experiment was done with Hershey and Chase. Okay, who's the two scientists? Hershey and Chase. This is the third experiment which is done to understand whether the DNA or protein was the exact genetic material. Okay, so first Griffith has done the experiment. In that experiment, it was clear that transforming principle is present. Okay, in the second experiment with MacLeod and McCarthy, it was very clear that DNA is the genetic material. And if you are considering the third experiment, it is Hershey-Chase experiment. Okay, what does this Hershey and Chase has done? Okay, not Chase. Okay, it is Hershey-Chase. Hershey-Chase experiment. See, Alfred Hershey and Martha Chase. They are two scientists. Okay, they are two scientists. We can put a colon here, hyphen here. Hershey and Chase are two different scientists. They have done the experiment or they have proved the DNA as the genetic material. Okay, so how they have taken or how they have proved DNA as the genetic material? They worked with the organisms known as bacteriophage. Okay, you have to understand what is bacteriophage. Okay, what is actually bacteriophage? What is actually bacteriophage? See, if this is a bacteria, if this bacteria is infected with a particular virus, okay, a virus that can infect a particular bacteria. Okay, bacteria is known as bacteriophage. Okay, so what is bacteriophage? A virus. Actually, it is virus. And what is the speciality of the bacteriophage? This virus can infect on bacteria. Okay, so they have done the experiment with bacteriophages. Okay, they have done the experiment with bacteriophages. Okay, so there is genetic material inside the bacteriophages. And if you are considering the structure of virus, see there will be there will be a genetic material, and over this genetic material there will be a protein coat. Okay, there will be a protein coat. This protein coat won't be entering into the host organism. You have to think that, okay? The protein coat will stay as light and protein coat won't be entering into the host organism. The genetic material, usually it will be RNA. Okay, usually it will be RNA. The genetic material will be moving into the host organism's body. Okay, so those viruses which infect bacteria is known as bacteriophages. So what or how they have done the experiment? Okay, so they have chosen the bacteriophages as the organism. Then they have treated it with different radioactive, what we can say, okay, radioactive phosphorus and sulfur. Okay, they have treated it with radioactive phosphorus and sulfur. So see, this bacteriophages they are treated with radioactive sulfur. Okay, they are treated with radioactive sulfur. Here, this bacteriophages are treated with radioactive phosphorus. Okay, so what is the speciality of the sulfur and phosphorus? Can you see phosphorus inside the DNA? Can you find any phosphorus inside phosphate group inside the DNA? Can you find? Yes, right. When we have discussed with the structure of nucleotide, we have seen that phosphate group is present, and this phosphate group will be there. Phosphate group is attached to the fifth carbon. Okay, phosphate group is attached to the fifth carbon, right? So we can say this phosphate group, radioactive phosphorus group, will be integrated with the DNA molecule. Okay, that is the basic concept you have to keep in mind. Radioactive phosphorus will be integrated with DNA molecule. Okay, so if you are considering radioactive sulfur, okay, radioactive sulfur, if you are studying the structure of protein, different amino acids are present, right? A long chain of amino acid will be forming a protein. Okay, that you have studied in grade 11. A long peptide bond or long peptides. Okay, amino acids which are connected with peptides, amino acids which are connected with peptides will form protein. Okay, so how does this protein, or if you are considering the structure of protein, there is sulfur molecules. Okay, sulfur. If you are considering structure of amino acids, what is there? Sulfur will be there. So this radioactive sulfur can integrate with protein molecules. Okay, if bacteriophages are grown inside the radioactive sulfur containing medium, that particular medium can give the radioactive sulfur to the protein molecules. So we can say that integration of protein molecule, integration of radioactive phosphorus molecule into protein is possible. Okay, so they have labeled it with both phosphorus and radioactive sulfur. Okay, then what happens? Actually, I told that they are bacteriophages, right? So they allowed it to infect. Okay, the second image shows that they are allowed to infect. What happens when it is infected? See, the black color genetic material is moved into the cell, right? If you are looking into the next, see, if you are looking into the second set of experiment, okay, this red color genetic material is moved into the cell. That means radioactive labeled phosphorus is entering into the host organs. Okay, into the bacteria. Okay, so again what happens? The radioactivity is moved, the radioactivity is removed. Okay, and if you are considering the bacteria, the genetic material enters. Okay, the genetic material enters, right? The genetic material enters. See, you can find black genetic material here. Okay, that means after this infection, they have centrifuged this. Okay, centrifuged the cells. Okay, centrifugation means what is centrifugation? It's a long rotation. Okay, rotation of different cells. So after centrifugation, what they got? See, if you are considering a test tube, you will get a pellet. Okay, you will get a pellet and you will get a supernatant. Okay, you will get a pellet and you will get a supernatant. So if you are considering the pellet, there contains the DNA. Okay, since protein is very lighter, see, protein was present as the supernatant. Okay, and when they observed, they find that the supernatant protein contains the radioactive sulfur. Okay, supernatant protein contains radioactive sulfur. That means inside the bacterial DNA. Okay, inside the bacterial DNA. This DNA is actually of bacterial DNA. Okay, they are infecting the bacteria, right? They are infecting the bacteria. So when they look into the DNA, there was no radioactivity, which clearly indicates that no DNA was transformed. Okay, no DNA was transferred into the bacteria. No DNA was transferred into bacteria. No DNA was integrated with the bacteria. Okay, so if you are looking into the second set of experiments, see, this is the second set of experiment. If you are looking into that, see, radioactive phosphorus, this is labeled DNA, right? This radioactive phosphorus I told that phosphorus, radioactive phosphorus will be integrating with DNA. Okay, since phosphate group is present in DNA, it will be integrating with phosphate. Okay, so that is actually going, the genetic material is moving from bacteriophage into bacteria. Okay, bacteriophage into bacteria. Then see this bacteria blending means the mixing or the integration of DNA, bacterial DNA with the bacteriophage DNA. Okay, second step is blending. It is also done. Third step is centrifugation. As I have told, here also what happens? Centrifugation has done. Okay, centrifugation has done. So if you are considering centrifugation, as I have told before, if you are considering the centrifugation, see if this is a test tube, you will get a pellet and also you will get a supernatant. Okay, pellet means a dense region, and you will get a supernatant region. Okay, so what is the speciality of this dense region and this supernatant? When it was checking, the supernatant was not containing any kind of radioactivity. Okay, any kind of radioactivity was not present in the case of supernatant. Okay, the whole radioactivity was present inside the bacterial DNA. Okay, bacterial DNA. That clearly indicates the DNA. Okay, see, this is radioactive labeled DNA. This radioactive labeled DNA is transferred to the bacterial cell. Okay, radioactive bacterial DNA is transferred to the cell. Then it multiplies and integrates and multiplies. Okay, when it has centrifuged, what happens? We got a pellet and we got a supernatant. Okay, when it was checked or when it was analyzed, what was the result? The radioactivity was not present in the case of the supernatant. Radioactivity was not present in the supernatant, but radioactivity was present inside the bacterial DNA. That clearly indicates the only material which transfers to the bacterial cell was DNA. That clearly says DNA is the genetic material. Okay, DNA is the genetic material. So this is the three different experiments done by different scientists, and this three different experiments is very important, especially if you are considering the Hershey-Chase experiment. Okay, in exam, in most of the times, usually CBSE has asked different question over the first experiment, Griffith experiment and third experiment. So you have to be thorough with first experiment and third experiment to prove that DNA is the genetic material. When you are writing the answer, you have to be clear with your ideas or concepts. Okay, what is the different organism? First you have mentioned the scientist name, then you have to mention what is the organism they have used for the experiment. If you are considering Griffith experiment, Griffith has used Streptococcus pneumonia as the bacteria for his experiment. Okay, then also how he has done his experiment, write in a crisp manner. If it is given for 3 marks, if it is given for 5 marks, you have to add or you have to write in a paragraph. Then you have to cut short in a small what we can say in a table. Okay, you have to include all the points in a table, then you have to explain about that. Okay, if you are considering the third experiment, okay, that is Hershey and Chase experiment, usually the CBSE has a tendency to give the question from this portion too. Okay, so be thorough with Hershey-Chase experiment. That diagram is very important. The diagram which is given in your textbook is very important. They may be asked this question with case-based study also. It can be integrated. Okay, so that is all about the 3 experiments which we have learned for the DNA as the genetic material. Okay, 3 experiments. Once again, it was with Griffith. Okay, first one it was with Griffith. Second one it was with MacLeod and McCarthy. Third one it was with Hershey and Chase. Okay, very important, dear students, very important. So next we will be moving into the next topic of this chapter, That is about the DNA. Okay, so in the previous class we have learned about DNA, right? What is the speciality of DNA, or what is actually DNA? DNA is deoxyribonucleic acid. Okay, what is DNA? Deoxyribonucleic acid. And you know about RNA, right? RNA, what is RNA? Ribonucleic acid, right? See, what is the difference between deoxyribonucleic acid, DNA, and RNA? See, one is deoxyribonucleic acid and second one is ribonucleic acid. Okay, ribonucleic acid. So if you are considering the structure in the 2 dash carbon, okay, in the second carbon there is no oxygen, no oxygen. Okay, there is no oxygen. That is why it is known as deoxyribose. Okay, but if you are considering RNA, in the second carbon, what is present? Oxygen is present. Okay, since oxygen is present, this can be very reactive. Okay, it can be very reactive. Since O group, oxygen is present, it can be very reactive. Okay, so it is actually not that stable. Okay, not stable if you are comparing with it with DNA. Okay, so that is the basic structure of DNA and RNA. And you know the DNA is double stranded in structure. Okay, so after learning all these experiments, you have to come across with replication process. Okay, what do you have to come across with? You have to come across with a replication process. What is replication? Replication. Simple meaning of replication is duplication. Okay, what is the simple meaning? It is duplication. Replication, replicating, replica. Okay, it is duplication. So, what is actually replication? Replication refers to duplications. Okay, then nucleic acid, DNA and RNA, they have the ability to duplicate due to the base pairing and complementarity. See, if you are considering a simple DNA, this is a DNA, right? If you are considering the DNA, okay, DNA is double stranded in its nature. Okay, double stranded in its nature. So we can say there is complementarity. Adenine pairs with guanine, guanine pairs with cytosine, cytosine pairs with adenine, adenine pairs with guanine. Okay, in the previous class we have discussed about that. Okay, so next one more molecule is there, biomolecule is there, and that is protein. Protein is made after the translation, but this protein is not having any kind of duplication. They won't undergo any kind of duplication. Duplication will be done only with the help of DNA. Okay, so again the scientists have worked on duplications, how the duplication have been done. Okay, so this is done by another scientist called Michelson, Matthews and Michelson. Okay, this Michelson-Stahl experiment, Matthew Michelson and Stahl experiment, it is also an important experiment in your textbook that you have to learn. Okay, so in total you have to learn 4 different experiments in this particular chapter. These 4 experiments are very important. You can expect at least 1 experiment in your board exam. Okay, 1 experiment you can, for sure you can expect. Among that, the 3 is very important. The second one is not that important. Okay, so fine. So what does they have done? They have done the experiment with nitrogen. Okay, radioactive nitrogen and radioactive nitrogen N15 and N14. Okay, N15 nitrogen and N14 nitrogen. Okay, N15 and N14. Okay, so what does they have done? They have taken the DNA. They have taken the DNA which is grown in N15 medium. Okay, in which medium the DNA is grown? The DNA is grown in N15 medium. After 20 minutes, what will be happening? Okay, this N15 DNA, see, if you are considering E. coli, okay, the organism chosen here is E. coli, Escherichia coli. Okay, this E. coli was allowed to grow in a medium which contains N15. Okay, the medium was N15. Okay, then later on it is centrifugated with the gravitational force. Then they shows a heavy band there. Okay, what they shows? what they shows? They shows a heavy band there. Okay, so next after this N15 medium, okay, after this N15 medium, uh, at first it was grown in N14 medium. Okay, at first it was grown in N14 medium. Okay, at first it was grown in N14 medium. From N14 medium it was taken and it was grown in N15 medium. Okay, so what is the organism? Organism is E. coli. E. coli. Okay, then this E. coli is grown in N14 medium. Then later on it was taken and it is grown in N15 medium. Then what they have observed? What they have observed after 20 minutes. Okay, after 20 minutes, what they have observed? See, the normal time period for replication in prokaryotes is 20 minutes. Okay. So they have chosen this E. coli as the organism. After generation 1, they have seen the DNA replication as this N15 medium DNA and also the N14 containing DNA was present. Okay, N14 and N15 was present. Okay, so let's write it. So see, they chose which organism? They chose E. coli as the organism. Okay, so we can consider E. coli, and this E. coli was allowed to grow in N15 medium. Okay, E. coli was allowed to grow in N15 medium. So the DNA will be having N15. The DNA will be having N15 DNA. Okay, then later on, the second session, what happens? This DNA containing E. coli was taken and it was grown in N14 medium. Which medium they have grown? N14 medium. Okay, so E. coli was grown in N14 medium. So we can put the color as, for N14, it will be the color. Okay, so this is for one generation. If you are considering E. coli which is grown in N14 medium, they will produce this color DNA. Okay, then later on this N14, the E. coli which was growing in N15 medium was transferred to N14 medium. Okay, it was transferred to N14 medium and allowed for infection for 20 minutes. Allowed the growth for 20 minutes. Okay, so what happens? This DNA will be replicated. The DNA will be replicated as like this. Okay, so N14 medium is also containing the DNA. So it will be also replicated. Okay, it will be also replicated. Then see, next in other 20 minutes, again it was grown in the N14 medium. Okay, again it was grown in the N14 medium. So what happens here? Here the DNA, the base strand will be white strand. Okay, so again this replicates. Again this replicates. Okay, again this replicates. See, it has 4 parental strands. Okay, it has 4 parental strands. It is present in N14 medium. Where it is present? It is present in N14 medium. Since it is present in N14 medium, what it will be showing? It will be giving the N14 DNA. Okay, it will be giving N14 DNA. Okay, so see, when it moves on, the parental strand will be moving. N15 will be exactly like that, and N14 will be adding. Okay, so that clearly indicates that the mechanism is semi-conservative. Okay, the DNA replication is semi-conservative. Okay, so we can say this is N15 medium DNA and it is given in N14. It is infected with N14. So there is a hybrid formed. And this hybrid, okay, there is a hybrid form. See, there is a hybrid form that is N15 and N14. It gives in between. The N15 medium, if you are considering the gravity, the centrifugation, see the band formed is in the in the lowermost region. Okay, but if you are considering the DNA grown in N15, N14, it was in the middle region. Okay, then later on it was added with N14 medium. Okay, generation 2 was also done in which medium? N14 medium. So N14 DNA will be present and the hybrid is also present. Hybrid will be in a little amount. Okay, hybrid in 50%, then N14 medium in 50%. If you are allowing this to grow in generation 3, okay, if you are allowing this to grow in generation 3, okay, generation 3, what happens? What happens? The N14 DNA will be multiplicated. The number of N14 DNA will be more and the hybrid will be less. Okay, it moves on. And this, every single conclusion of this experiment gives that the DNA replication is semi-conservative. DNA replication is semi-conservative. So, semi-conservative mode of replication is actually explained by which experiment? Michelson and Stahl experiment. So, this is the 4 important experiments that you have to study for your board exams. Okay. So, next we are discussing with replication. Okay. What is replication? As we have mentioned, what is replication? Okay. DNA replication. Okay, what is DNA replication? Replication is formation of replica. Okay, formation of replica. So, DNA should be there. Okay, what is the essential requirement? Sorry. First one is DNA itself. Okay, second one, for DNA replication, we need different enzymes. Okay, what is needed? Different enzymes are needed. Okay, different enzymes include DNA polymerase. Okay, DNA polymerase. DNA dependent DNA polymerase. Okay, second one, it is important as DNA ligase. Okay, so we can say DNA polymerase and DNA ligase is the important enzymes which is used for replication process. Okay, and then we can see what happens during a replication. Okay, what happens during a replication? Consider a bacterial DNA. Okay, consider a bacterial DNA. If you are considering a bacterial DNA, it will be circular in structure. Okay, it will be circular in structure. So, this circular structure of DNA, it is in a circular mode. So, if you are considering the circular mode of DNA, the replication has to be started. Okay, see, it has double stranded, right? So, we can represent like this. Since it is double stranded, we can represent like this. Okay, it is double stranded, it is circular in structure. So somewhere this DNA replication has to be started. Okay, the point at which DNA replication start is known as consider this is the particular point. Okay, this is the particular point where replication starts. So this particular point of replication where it starts is known as Ori region. What is the region name? Ori region. What is the meaning of Ori? Ori means origin of replication. Okay, origin of replication. Okay, it has hydrogen bonds. So this hydrogen bond has to be removed. As we have studied, all the base pairs are connected. Adenine pairs with thymine with 2 different bonds. Then guanine pairs with cytosine with 3 different hydrogen bonds. So hydrogen bonds are very important. This hydrogen bond have to be broken down. Okay, so this origin of replication is the particular site or particular region where the DNA starts replicating. Okay, so if you are considering this particular region is just opened, see, when it starts replication, this replication will be forming a Y-shaped structure. Okay, it will be forming a Y-shaped cork structure. Fork structure, not cork, it is fork structure. Okay, see, I'll draw the structure here with another color. See, this is the DNA, double-stranded DNA, and this is the origin of site, and it forms a replication fork. Okay, it forms a replication fork. So if you're considering the replication bubble, this is the replication region, starting of replication, you can find it as the replication or replicating fork. Okay, we'll call it as replicating, Y-shaped replicating fork. In the both sides, the replication fork will be present. Okay, once it is unwidened, okay, what happens? Once it is unwidened, the polymerase enzyme will be starting acting upon them. Okay, so unwidening is done. After this, what happens? The polymerase enzyme has to be done. Before that, a primer has to be added. See, if this is considered to be primer, always the synthesis will be done in 5 dash 3 dash direction. This is 5 prime, 3 prime, this is 5 prime, this is 3 prime, and this is 5 prime, and it will be 3 prime, then 5 prime, 5 prime, and 3 prime. So this is 3 prime, and this will be, okay, this is 5 prime, and this will be 3 prime. Okay, so always you have to keep in remember the replication starts from 5 prime to 3 prime. Okay, so this will be the 5 prime to 3 prime. If you are considering the DNA, synthesis will be in 5 prime 3 prime direction. Okay, here the synthesis will be in 5 prime 3 prime direction. This is, sorry, it is written as 3 prime, right? Okay, so this is 5 prime and this is 3 prime. So always think that the synthesis will be in 5 prime and 3 prime direction. Since it is having a 5 prime, it will be having Okazaki fragments. Okay, so this will be 5 prime and this will be 3 prime. Okay, so always think that it is done with the help in only one direction. Synthesis will be in one direction, that is 5 prime 3 prime direction. Okay, DNA replication will be done only in one direction, that is 5 prime 3 prime direction. Okay, and the two strands will be acting as the template strand. The two strands will be acting as the template strand. One strand will be known as continuous strand. Okay, one strand will be known as continuous strand and the other strand will be known as discontinuous strand. Why it is known as continuous and discontinuous strands? See, you can find this is the replication process. Okay, after replication, you will get the result as like this. So, what is the enzyme which helps in the replication? At first, primer has to be added in the 5 prime direction. Okay, what has to be added? Primer has to be added. What has to be added? Primer has to be added in 5 prime direction. Why primers have to be added? Why primers have to be added? Primers are actually small part of RNA. Okay, unwidened. Okay, the DNA will be unwidened with the help of an enzyme. Then primers will be added with the help of primase enzyme. Okay, then after that, what happens? Synthesis have to be done. Okay, different DNA synthesis have to be done. This DNA synthesis is done with the help of which enzyme? The major enzyme of this DNA replication is polymerase enzyme. Okay, what enzyme? Polymerase enzyme. Okay, then DNA helicase, which helps in the unwidening of DNA. If origin of replication is identified, DNA helicase come and acts upon the DNA. It will be opened. It will be widened. Okay, so then polymerase enzyme comes. Add the polymerase, add the DNTPs, then it will be acting. Then finally, Okazaki fragments is formed. Okay, in the discontinuous strands is formed, and the Okazaki fragments have to be ligated. For that, what happens? DNA ligase will be coming and acting upon DNA. Okay, Okazaki fragments are joined with the help of DNA ligase. These are the three major enzymes used in DNA replication. So, if you are considering DNA replication in prokaryotes and DNA replication in eukaryotes, the enzyme will be little different, but the process will be the same. First, initiation, then second one, the formation of replication fork, then third one, it is the ligation of discontinuous strands. Okay, so you understand about DNA replication? It is important, okay? DNA replication. Here you have to stress on different enzymes which is used for DNA replication, and about the replication fork, the Y-shaped fork. You have to be thorough with the structure of replication fork, how it will be coming and how the synthesis will be taking place. It will be always in 5' and 3' direction. That is the important points that you have to keep in remember. That is the questions in for exam point of view. Okay, so that's all for today's class. We'll be continuing with the next session with this transcription and translation in the next coming class. So before that, Danya ma'am is waiting for you. Danya ma'am will be joining you with November 11 on this coming Saturday. Okay, until then, take care. Bye bye.